So as been said, this is something that's really come out of those unusual symptom reports, and it's really focused on trunk diseases. Kiwifruit trunk diseases have really been known for at least 20 years, but over that time they've been increasing in incidence and in visibility. They've variously been referred to as vine decline, vine decay, swollen trunk disorder, crown decay disorder, and kiwifruit wood decay. And the symptoms are varied as well. We've got the crown decay, swollen or uniformly enlarged trunks, discrete swellings of trunks or leaders, which show up as bulges and cracking, full-on cankers, sparse canopies, dieback, and of course sudden death. And when you cut those trunks open, you'll quite often see discoloured areas within the wood. So at the time, we were calling this vine decline, and so we started a project at the beginning of last year called Emerging Risk of Vine Decline. And this was really to discover which pathogens were associated with that vine decline. And now at the request of Zespri and KVH, we focused on the fungal group Neonectria, which largely consists of fungi with anamorphs, asexual states of Fusarium and cylindrocarpin types. Now, overseas, this group of fungi has already been associated with wood decays of kiwifruit, and previous work in New Zealand has also indicated that this group is likely to be involved in vine decline here. At the same time as this, the Neonectria canker was starting to appear in Motueka, so we were having unusual symptoms down there as well, and that actually falls into the Neonectriaceae group too. So what we did is we went out and we surveyed one block on each of 3 orchards. That was in Paengaroa, Te Puke and Motueka. And we walked up and down the rows and we assessed the amount of visibly diseased vines, and we also marked what kind of symptom we were seeing on each of those vines. We went back and we sampled the trunks of 10 asymptomatic and 10 diseased vines, and we took samples from the bark at the base of the vines, and we took woody cores across the width of the trunk at 30 centimetres above the ground and at 1 metre above the ground. And in the case of Motueka, we also sampled right through those leader cankers as well. Now, taking isolations across the length of those woody cores actually gives us a good idea of where those fungi are actually showing up across the width of your trunk. Are they coming in from the outside, or do they look like they're moving up and down the centre? So how prevalent was it? Between 18 and 34% of the vines were visibly diseased in those 3 blocks. And of the healthy vines, 2 to 22% appeared to be replacement vines. We didn't include, there was a lot of gaps in the canopy. We didn't include any removed vines because although we knew some of those were removed due to disease, some of them were just actually removed due to planned turnover. So the true incidence is actually likely to be higher than what we're reporting here. In addition to that, asymptomatic vines, they often had stained core samples indicating early disease. And what you're seeing at the bottom there, that thing, just trying to figure out how everything works, that's actually a core sample across a trunk. And you can see there's big stained areas on it. And this one, that actually related to Neonectria. So symptom incidence, not only did the blocks have different, the blocks did have different main symptoms. In Orchard 1, we were seeing a lot of swollen trunk, so that big fat trunk, and a lot of collar rot. In Orchard 2, it was mainly the collar rot, and down in Motueka, in Orchard 3, it was cankers and the swelling and cracking of the leaders that seems to be the precursor to the cankers. Right. Bringing back our samples to the lab, doing the fungal isolations, what did we actually find? We found that not only each orchard block was different in the most common symptom, but also in the most common species that was found in each orchard. There was a lot of overlap between the 3 blocks. Now, for those of you who have never seen fungi in a plate before, all of these beautiful pictures here are different species of Fusarium that we have recovered from those orchards. And while they look really pretty in plates and they have some beautiful colours, they don't look so good on your orchard. So So we found there was many species within the Nectriaceae in these 3 orchards. So we've got a great big list of different things there, a lot of Fusarium, Ilyonectria, Neonectria, and so forth. But 3 major groups were more prevalent in those diseased vines, and that was Neonectria microconidia, the Fusarium solani complex, and the Ilyonectria species group. Neonectria microconidia, probably better known now as Neonectria canker of kiwifruit. It's a very close relative of Neonectria dotissima, which causes European canker of apples. We found this fungus in all 3 orchards, not always associated with symptoms. Sometimes it just appeared to be hanging out in the trunk. However, it was highly associated with those cankers down in Motueka. And we also know that it causes fruit rots of kiwifruit, so I guess we know where the inoculum is now coming from. Now, just a little bit about these pictures here. The bottom left one there, that's one of the cankers from down in Motueka. These lovely red blobs are perithecia that were found in one of those cankers, and that's one of the spore-forming structures that you see with this organism. Top right, this is a gold 3 canker that was from up in Kerikeri, and when that was cut across, what you're seeing is this really ugly wood in the middle there. Naturally, this was dying above the canker. Ilyonectria species, not quite as pretty. We found 4 Ilyonectria species within the 3 orchards, and they were Ilyonectria europea, Liriodendri, robusta, and torosensis. Torosensis we only found in 1 vine in 1 orchard. Liriodendri we only found down in Motueka. Europea and robusta we found in fairly large numbers in all 3 orchards. Now, Ilyonectria is probably, as a genus, is probably better known for causing blackfoot of grapevines, and there is an overlapping set of species between the species set that is causing blackfoot of grapevine and the set that we're actually finding here. So some of those are the same thing. With Ilyonectria, we don't know whether they're pathogenic, we don't know whether they're causing symptoms or whether they're coming in later. There's no information on that. And they do seem to be a bit random. That being said, this fellow here is the only one that I've actually found that is Ilyonectria all the way across that symptom. Fusarium solani. This one's a bit of a worry. We found it in very high numbers in all 3 orchards, but it was the major group of the diseased vines in orchard 2. And if you remember, that's the one that had the high numbers of collar rot. It's a well-known pathogen, and it's been found in hundreds of different crops around the world. And we have previously found it in New Zealand, associated with sudden death of vines in several orchards. And in those cases, it tends to be the major organism in the cross-sections of the trunks in those vines. In this project, we've found that there's a high level of genetic diversity within the group of Fusarium solani isolates that we're finding. And this, down the side here, is a phylogenetic tree, looking at the genetic diversity. For those of you that are not familiar with these, imagine it turned sideways. It looks very much like a family tree. And the closer these organisms are on a certain branch, the closer they are genetically. It's like your brothers and sisters or your cousins. And what you're seeing here is that there's 3 major groups of more closely related isolates. They're not just genetically different, they're actually morphologically and behaviourally different. This big red group up the top here are the only ones out of all of those that actually happily form perithecia in culture. And this organism forms very similar red perithecia to Neonectria. And the blue lot in the centre here, they were only found in orchard 2. So obviously the next thing that we would do in this project would be pathogenicity testing. All of the fungi that we've found in this project there's no information on whether they're pathogens of kiwifruit or not. Are they opportunistic? Are the fungi causing or contributing to the disease? And which ones are the most aggressive? Which ones should we worry about the most? So this uncertainty is currently being addressed in other projects for Neonectria microconidia and for the Ilyonectria species group. Pathogenicity testing is already in progress for these ones. Testing does need to be done across the range in the Fusarium solani complex. And as I've said, there's a wide genetic diversity there. Generally, that means that a population is more adaptable and it can be more difficult to control. So, kiwifruit trunk diseases, KTDs as we like to call them. They're a complex problem. They're likely to be caused by a complex of fungi. They're actually very similar to grapevine trunk diseases, which are also caused by a complex and changeable group of fungi. And there is some overlap between the species in grapevines and the species that we're finding now in kiwifruit. They, however, have had 100 years of research on theirs, and they are still increasing in incidence. And there's still a limiting factor for grape production, not only worldwide, but here in New Zealand. So we are barely scratching the surface of what is happening in kiwifruit. It really needs research to unlock the different fungal combinations involved, the sequence of infection, and the source of inoculum, what is coming first, where is it coming from, the species epidemiology for each of these organisms. We also need to look at the other fungi that are not in the Neonectria that we're not focused on in this project. For example, Neobulgaria alba was actually what was actually what was found from side to side in this one up here. These are from a different project. I didn't go around chopping people's trees down. Also, the effect of cultivar, rootstock, orchard age, and the environment. And hopefully all of these things will give us points of intervention and ideas of how we can actually control these. So other questions that have been started to be answered in this project were really, what have we actually got? What is in our orchards? What is our current biodiversity? And that will help us with what is new, when is something an incursion, when is it just something that we've seen before or have seen a little bit before and it's increasing in incidence. Moving on to what to do about these things, kiwifruit trunk disease management. Not a lot is known, but this is the current KVH recommendations. Regular orchard monitoring. So vines with unusual symptoms should be tagged and avoided when you're conducting orchard practices that involve open wounds. It is unlikely that seriously affected vines can be cured. They've probably been infected years, if not a decade ago, with some of these pathogens. These vines should really be removed and replaced and a replanting strategy developed. Having said that, in the case of maybe Neonectria canker, they may be able to be managed by removing the affected part of the vine well below the symptom and redeveloping a replacement leader, and that may stave it off for a while. Obviously, tool hygiene is key. Sanitise tools as often as possible, ideally between vines, but between rows should be routine. Always sterilise your tools really well before entering a different orchard. And the use of wound protectants containing fungicides really should be used to protect wounds whenever possible, particularly in those orchards where there is evidence of vascular disease, and particularly after vines that obviously have something wrong with them. And report the unusual. So pretty much that comes down to monitor, cut out or cut back, sanitise, protect, report. And thank you to Zespri and KVH for funding this, the growers and orchards managers for putting up with us, and Kieran Mello and Kai Lewis from Plant and Food, who stomped up and down orchards with me, and Hayley Ridgway and her team for doing many of the Ilyonectria identifications. Thank you.
Any questions on that? For and consultant, when you're replanting, do you have a recommended sort of minimum distance, or what's the suggested protocol about re-establishing with new plants?
I'm happy to give that one a go. I was gonna go, but not in the same hole.
I'd Linda up, but she's avoiding eye contact.
She just looked away.
I think all I can do, Tim, is reference advice that I had from the late Mike Muller, and it's very much around resting the area that you've immediately come up with, trying to, move to the side. Obviously, you need to do that within your existing pattern. It is pretty tricky. We don't know the exact distribution of these things, so at least getting out of the general area is a good idea. Jo, I don't know if you want to add anything to that.
I would go with that.